Okay, I think we can continue with our next speaker. It's Dirk Petersen. He's the founder and executive director of SECO International, a coral conservation restoration organization based in the USA and in Germany. He initiated SECO and the coral breeding approach during his PhD project, which was done in collaboration with the Rotterdam Zoo and the University of Duisburg-Essen, I think. SECO is a global network of scientists, public aquarium professionals, and local stakeholders, fostering multidisciplinary strategies combining research, education, outreach, and active reef restoration for the conservation of coral reefs. Together with the Californian Academy of Science, the Nature Conservancy, and partners of various disciplines, SECO has recently launched a global coral restoration project to advance science, technology, and application of innovative coral restoration techniques. And he will give us an introduction what he is doing and how coral reef restoration will take place in a changing environment. So, welcome.
Thanks, Marlene. It's great to be here today and to add to this discussion. It's really great to have those other speakers this morning that come from a very different background. And actually yesterday, the coral people, we have discussed a little bit and how deep do we have to go into what corals are. So I assume, I hope that everybody knows a bit about coral reefs and has some background knowledge. This coral here is the Alcon coral Acropora pomata that you only find in the Caribbean, which is a key reef builder, a very extremely important species that only occurs in shallow, high wave exposed areas, which is a very important coastal protection and creates habitat for other species. Unfortunately, this species has been one of the two first listed corals as highly or critically endangered under the American Endangered Species Act and the IUCN Red List. More than 95% of the species have disappeared throughout the Caribbean. And if we look into the other side of the world, to the Great Barrier Reef, these pictures have gone around the world this year and last year, and I guess everybody knows now what coral bleaching is, and eventually people don't know yet what corals are, or that corals are animals, but nevertheless they know that coral reefs are highly endangered, or scientists, I think or the science community, I guess, was not surprised about what was happening in the Great Barrier Reef, but actually shocked, I guess, about the dimension and the expansion of the bleaching that happened in the Great Barrier Reef in the past 2 years. And if you look at the scale here, these are hundreds of kilometers, and so if a coral bleaches, it does not mean that it automatically dies. They can really recover well, but we can assume that a few hundred square kilometers of coral are lost because of the past 2 years and the global bleaching event. And personally, I was shocked to see this picture. If I would know better, if I would not know better, I would think this is a Caribbean reef. But this is Lizard Island in Australia, and I was there a few years ago, and it was a very pristine reef, and now it looks, or parts of it look like this. You have to know that if bleaching occurs, and if corals don't survive that, they die, and what remains is the skeleton. And very often these dead pieces of skeleton there are recruited by algae, and any new coral settlers, they have a hard time to settle on these surfaces and to survive and to compete with these algae. This is the typical situation, not typical, this is a very extreme picture here, but the situation in the Caribbean has been a bit different compared to areas like the Great Barrier Reef, where and the Great Barrier Reef is good management in place, but the bleaching that happened, of course, came from global effects. Whereas in the Caribbean, a dramatic decline started in the late 70s, early 80s, and there are various reasons for that. But what happened was, and this is a really famous example from Jamaica, where you could observe as the coral cover decreases, the algae cover increases, and we call this a phase shift from a coral-dominated to an algae-dominated ecosystem. And from a restoration point of view, this is a major. I guess this is impossible. Unless you would really remove manually these algae and then at the same time cover this area with a huge biomass of coral and then introduce the grazers that are missing to control the algae. I guess this reef is lost. However, there are many reefs that are in an intermediate phase from a coral cover or high coral cover to this situation, and there is hope to eventually reverse that. However, this does not happen without a cause. So there is eutrophication, there's overfishing, there are diseases. There was this massive disease outbreak in the late 70s, early 80s that caused a massive die-off of diadema, of a very important grazing sea urchin. And yes, and this is Fort Lauderdale, sewage pipe, and people who dove there told me that it's not as bad as it looks. I don't know. I just think there's a lot of funding, or there's a lot of activity in Florida and Florida Keys to do restoration. But I guess as long as this is, for me, it's just impossible. I mean, to have this situation there and to do all the restoration, there's no use. So this has to change first. So I told you already about the very important Alcon coral and the Staghorn coral, Acropora cervicornis. And overall in the Caribbean, with the die-off of the diadema, of these very important grazers, we could observe this drastic decrease in coral cover overall in the Caribbean. So together with the very recent event in the iconic Great Barrier Reef, it is clear that we have to look into new directions for management and conservation. And the questions I address today is, how can we restore coral reef resilience at scale? So very important is to keep in mind what is the goal of restoration. What we need are diverse coral communities that are able to persist in the changing environment. That means it doesn't really help to keep on introducing and outplanting propagated coral every year again and again. No, we have to, the goal is to have self-sustaining populations at some point. And we believe we can manage this eventually, or we have the greatest chances of success if you look at an array of techniques to cover a very wide range of different scenarios, and not to focus on single or one technique in one direction. So it's really very important to look at any direction that might be promising. If you want to restore coral resilience at scale, reduce CO2 emission and enhance local management, like the situation there in Florida. There's no doubt this is the priority. This will always be the priority. And the press, when we give interviews, the question is always, oh, now you've solved the problem. Now you can restore coral reefs. So, and I think Elle told me the other day that there was a headline in the British news now with the recent publication from Pete Harrison. So now we can restore coral reefs and we don't have to deal with climate change any longer. This is really dangerous. I mean, this is it's the wrong direction, the wrong way of thinking. However, if we all know by now that just looking at the traditional management tools, it is not sufficient any longer. We have to look at active restoration as well. If you look at active restoration, we've set 2 priorities. It's scale of restoration activities, and then we're looking at this whole field, and that's why we're here today. Assisted evolution, human-assisted adaptation, acclimatization, synthetic biology, but also genetic diversity, which we should not forget. Looking at the scale of restoration, to give you some background, so corals can reproduce primarily in 2 ways: asexually, in a whole array of different ways, but the most important one for restoration is fragmentation, where you basically break off pieces of a coral, glue them manually on a substrate, let them grow in a nursery for a while, and then you outplant them, you transplant them on the reef. And here this is done with an underwater pneumatic drill and then epoxy cement. If you look at using sexual reproduction, so most corals, they release their gametes in a massive spawning event, and we use this to collect some of these gametes, fertilize them in the laboratory, rear larvae, and once those larvae are competent, we settle them on specific substrates. And this is done here with a group around James Guest. So here you have this nursery situation again, and then there's the outplanting, and again you use every single coral is manually attached on the reef. As you can imagine, this may work for a few thousand coral, but if you look at 50,000, 100,000, a million coral, this is impossible, because it takes a lot of labor, and the labor creates costs. The costs are huge, and this is why restoration currently is limited to a scale of usually less than a hectare if you look at a single project, whereas the scale of degradation is occurring at hundreds to thousands of square kilometers. So there's a big gap here, and the question is, how can we narrow down this gap a little bit or significantly enough to insert or to include restoration as a meaningful tool in the whole toolbox of management tools that are out there. So I want to give you like 3 possible solutions of hope. And please only look at this in the context that this is an addition to management, and this is an addition, the background that there's some reduction in CO2 emission and the local enhancement of local management. So an idea could be to culture larvae in large numbers. It's no big deal to culture 1 million larvae and more in a single spawning event, and then pump those larvae or introduce those larvae in a cage environment. So you force the larvae; they will not be dispersed, but they will be forced to settle on this area here. Here it's the same. It's done with a net, plankton mesh. And these studies showed there is indeed increased settlement, significantly increased settlement compared to the control. The study of Edwards et al. Showed that after a longer time of monitoring, there was no significant difference. So there was no additional recruitment compared to the control. So it did not work out in the longer term. Whereas the recent study of Dela Cruz and Harrison showed indeed, which is not shown here, but I can tell you, that there was a successful outcome of the study, which gives some hope. Another approach is, and you have to look at all these restoration directions and technology, like from a cost-benefit perspective. So it's very important, what are the costs and what is the outcome? If you introduce have a success rate of like 80%, and each coral that you introduce on the reef has a cost of several $100s per coral, it might be more effective to use a technology where you have eventually 25 or 10 or 5% success, but each coral is $1 in cost. The easiest approach you've seen with just taking larvae, settle them, force them to settle, and then they eventually survive. That would be great. Another approach is to go a step further with the intervention and to collect gametes from natural populations here or from a nursery environment, fertilize those gametes where you can do cross or specific crosses, eventually use more resilient coral genotypes eventually, and then we culture those embryos, and here you can attain like 90% fertilization and more. Usually it depends very much on the year, on the population, on the success, on some factors, but it's no big deal to get close to 100% fertilization rate. And then we are looking into mass culture those embryos in situ situation with these pools that have been that we have modified after Japanese colleagues that have used them already like 10-15 years ago, and we introduce substrates in those pools. So once those larvae are settlement competent, they can settle on those substrates right away. And then those substrates are designed in a specific way that they are capable or likely to self-attach on the reef because of their shape. So you get away from that very time and labor consuming transplantation process, which I haven't told you before. It takes about half of the whole labor that is needed for the restoration process goes into the outplanting only. So by reducing the time to outplant corals, you can save a lot of time and costs and enhance the efficiency of restoration. So here we just use, you see the settlers after the very short nursery period, and then they're batched simply on crevices on the reef, this is from Mexico. We have done this at other places. So this is also a direction we want to look into, and we're currently optimizing those substrates to fit different reef environments and situations. And then there is another direction where you use asexual fragments, we call them microfragments. And basically, if you look at a larger piece of coral, it's cut into smaller pieces, and then those smaller pieces are glued, or they're grown for some time, and then in a land-based nursery, and then they are glued on larger concrete pieces very closely to each other, and they will show an enhanced growth rate compared to if you would leave the whole piece in one piece as originally, which could help to overcome very slow initial growth rates of more massive corals. So now if we look at enhancing resilience of coral populations or at the ecosystem level, I just want to dig into this a little bit because there's little time, and there's a lot of expert talks in the afternoon. So if you look at this scenario where you had a phase shift towards an algae-dominated reef, you will not get far with using more thermal tolerant corals. And this is something we should not forget. If we get more thermal tolerant corals and that works out, they still would have to deal with this situation, or well, this is extreme, but you know, and this very initial life stage where we have to look at a larvae that is less than 1 millimeter in size and length turns into a primary polyp, the initial of a new coral that is less than 1 millimeter in diameter, you can imagine how easily they are outcompeted in this situation. So eventually we have to look at other species than those original species, like the alcon corals or the acroporas that are very fragile in these early life stages, and they will not make this. But we know from studies in Curacao, on observations, that this is I would say, an intact coral reef, and you find this in front of the capital of Curacao. And Curacao has about 130,000 inhabitants, and about 100,000 live in Wilhelmsted. So you would never expect to go in the water in front of this refinery and cruise ship harbor and all the buildings. And there's little to no sewage treatment that you would find any live coral, but you do. And what you find is not this original, like acropora, staghorn, alcon coral dominated coral reef, and other species. There's a shift towards these brain corals, because we found that they are doing better, even they have a higher survival if you introduce early coral settlers in a control environment, and then in environments that are eutrophicated, they do even better. So they might be, this group of species might be capable or helpful in any restoration effort to help to turn a algae-dominated reef in a coral-dominated reef again, and set the layer or the basis to then establish and recruit other coral species and get this area back to a coral-dominated reef and intact and functioning reef. So it's very important, besides all the looking at more thermal tolerant corals or super corals or whatsoever, to look also into, and not to look only into critically endangered species that have been in the focus of looking at the Caribbean, and there was a lot of funding available. No, it's also very, very important, we believe, to look in like species that you still find on the reef in high numbers, and that are capable to help to create in the reverse ecosystem functioning. Now, if we look at assisted evolution, I think it's very important to realize that, as far as I realize, that assisted evolution is always selection. So it means a reduction by definition of genetic diversity. So, no, okay, very good, very good, and I'm looking forward to that discussion. So, okay. So the question is, what do you go for? Is it you preserve genetic diversity, or, and ideally, you look for more thermal tolerant corals? Within our science advisory board, or our science advisory board advised us to look at the priority to preserve genetic diversity and to look into the potential of assisted evolution, and ideally have those two directions going with each other. So we are looking, currently we are looking at, whenever we do any of this work, to get a maximum of individual colonies together and to be cross-fertilized in what we call a batch culture to maximize genetic diversity. With this species, with these highly endangered species where we have lost more than 90% of the population throughout their range, you're losing genetic diversity. So any successful, and these corals, they don't reproduce in the wild any longer. Also, they spawn. Some of those populations, you don't find any significant numbers of juveniles. So by getting any individual surviving through these restoration efforts, you add to genetic diversity. And then, very importantly, I think, is cryopreservation. Here's Mary Hagedorn's lab, she's working since many years on this, and so it is possible to cryopreserve sperm. Hopefully it will be possible to cryopreserve eggs at some point. I think it's possible to cryopreserve tissue. And in the whole situation of coral reef decline, yes, it is a very important thing to start creating cryobanks and to preserve genetic diversity. But I'm not going to talking about potential trade-offs with more thermal tolerant corals, so that these thermal tolerant traits might be connected with reduced growth, for example, or reduced fecundity, how heritable those traits are. If you find those traits or with coral population at one place, does it work the same way at another place? I really want to look and give some ideas and some considerations and eventually add to the discussion today. And as CCOR is an intersection between science and application, I want to look at, okay, well, let's hopefully there is success in this assisted evolution idea, and we get the results, and what then? How would we apply those? How would we integrate this concept into restoration practice? And first of all, I think the most obvious question would be, okay, how do you know that the coral is more thermal tolerant as a practitioner? I mean, you can do all these, and we talked about it yesterday, how complex this whole field is. And so if you look at this picture, you think, okay, this is my more resilient, more thermal tolerant coral. Or this is a more resilient coral, worth to breed and to use for restoration. It could be that this individual here that is bleached is more resilient because it will recover this bleaching event and survive, whereas this one will be wiped out very soon by a disease. It's from a practitioner's point of view, and we had this workshop last year hosted by NOAA in Fort Lauderdale, and there were all these practitioners, and they are, they need a very simple recipe how to use this information and to translate this into action. So I've added this one here. Everybody knows that, I guess, or everybody who works with corals in the field, which is a very simple chart how to identify the health status of a coral colony in the field with the diving. So I think we need to think as we go ahead with the coral assisted evolution idea to have something like a comparable, very simple field kit that practitioners can use to do something with it. And then, yes, okay, once you have those coral individuals identified that are more thermal tolerant, what are you going to do with them? Do you leave them in the field? Well, I guess for these alcon corals, yes, you will, because they are 2 meters and more in diameter. Or you cryopreserve their sperm or tissue. Or do you have them in an in situ nursery, or do you keep them in a land-based, like here the CSIM flow-through systems and preserve them there? And probably, I guess, we're looking ideally at a combination of all these approaches just to spread the risk. But it's definitely important to start thinking about it, I think. And then, okay, I learned this morning it's not migration, but it's translocation. So translocation of genetic material is also an issue, I guess, to start thinking about. So so far we work very regional. So whatever we collect on genetic material, on gametes, we reintroduce where we have collected or in that area, which is usually then restricted to islands or coastlines of a few kilometers in in extension. But eventually, with this continuing coral crisis, we have to think about and the easiest way would be to use prior preserved sperm or larvae, because you can move large quantities from one area to the other, and to start thinking about, well, okay, how could we? And I'm really glad that it was partially addressed in the previous talk. So yeah, to take Florida, just name it, take Florida sperm and move it to Curacao, or vice versa. What are the logistical constraints here, permitting wise, and what makes more sense to reduce to use genetic material from a pristine reef, or to go to the Florida Keys where you find very little genetic diversity, but those genotypes, they made it, they survived, so maybe they're hardier. I'm very provocative here. So but I think it's absolutely the time to start thinking about this. Then yes, these efforts would need some great coordination and guidance, and we are frequently contacted by individuals or small communities that do restoration, and they ask for advice, and you can read then that they have no idea what they're doing. They want to help, and I'm talking about fragmentation, so gluing every single coral and getting it out, and then they probably talk about a few thousand, and they don't know what they're doing. So eventually they're causing more harm than they help. So now look at the potential of using sexual reproduction and larvae, where we are dealing with 1 million, and the harm you could do with that, including like, these interventions with assisted evolution. So you have to be, I think there needs to be a very strong guidance to direct those efforts, and then there needs to be a coordination at national level, most importantly at international level, because you probably look at the Caribbean as a in total, and then there are other areas where it's just international, yes. And then how do those practitioners know how to deal with these more thermal tolerant corals? So there needs to be training workshops, webinars like this one, manuals for practitioners. And then the technology that comes along with these ideas have to be made available with protocols to those practitioners. Before I end my lecture, I want to emphasize a project that we have just recently started with the Nature Conservancy and the California Academy of Sciences and many other partners, to address exactly some of those issues and questions that I've just raised before. So we are looking at moving the technology development and the knowledge forward for scaling up restoration efforts, looking at coral resilience to stimulate partnerships. This is really a global effort. I think a global crisis needs a global effort. Capacity building, and to implement then the knowledge. And that's going to be crucial. Again, if you know as a scientist how to deal with it, it does not mean that a practitioner can do anything with it. We have an initial focus in the Caribbean, and then depending on how things develop, we will expand to the Pacific and build partnerships there. The idea is to have capacity building centers in the Caribbean, which we are currently establishing. And those will serve as a hub for training workshops for practitioners, and to give guidance to those practitioners to implement those new technologies in their region. And that's it. Thank you.
Thank you for the now more practitioner's point of view, and I think at all ends we have to work and start to do something, and I guess it has to be at the same time. I think there are also questions regarding your work and your presentation.
Yeah, there's one.
Thank you for your talk. So you showed that there's evidence for shifts in species composition on a reef, perhaps from one type to another, perhaps because of environmental change, maybe that one. But in the Caribbean, the coral species are one of the dominant species. They're one of the key reef species, they're ecologically relevant. A lot of work is being done to conserve and restore them. But this past year, the hurricane season, I know that there was widespread destruction of the corporates. And so my question is, so I know that they are more fragile, they're branching, and we can also use this as a way of fragmenting dispersion to new areas. But if they are essentially less vertically proof than the still growing massive corals, and if there is going to be a shift from perhaps branching to the still growing massive corals, which will take time, you know, because the hurricane season is going to continue, right? You have hurricane season every year, and it seems that it's going to get worse as the intensity increases. So my question is, as a practitioner, in your opinion, would you start to question whether we need to continue doing so much focus on the branching species if they are really, in fact, going to be continuously updated every year for the hurricane season in this one month region of the world? Or would you say, no, let's keep going forward and do what we can before?
Yeah, the branching versus the massive species, they have a very different ecological function. And we are looking not so much into species conservation, but more into the conservation or the restoration of ecological functioning. So unfortunately in the Caribbean you have very little choice because you have very few branching species, and under natural conditions, or if those corals are very well adapted for hurricane disturbances. However, with all these other stressors going on, like the local and the global effects, yes, they have you know, usually they would break, or they do break during a hurricane, and we've seen that also in Curacao during heavy storms, that these huge colonies, they are just tumbling over, and then you have this 2-meter colony upside down the next day, and they reattach and regrow and continue. So no, I definitely think, yes, we have to continue with the branching corals, because the massive corals will not replace their role in the ecosystem.
So within your Caribbean framework, this novel project, so what is your decision and legal framework you now really anticipating, or are going to start to translocate?
No, we. Okay, no.
Okay.
We don't do that. No. It's an ongoing debate. I think like 10 years ago, Ilyana and I spoke, and we were like working in Puerto Rico and Curacao, and I said, you know, why don't we just take like some larvae from Puerto Rico and put them to Curacao, and vice versa? It might add to the genetic diversity and success. And no. So no, we don't do that. We are working at all these places. So we use the same technology. We use those places currently as research hubs where we get feedback, and then we can condense that and develop further. And all those places work independently. They collect locally gametes and do the whole work and reintroduce or outplant the resulting coral on the same reefs. So there's no.
Can I add, so what's your aspiration? Do you want to have that happening? And if yes, what's your strategy to eventually get there?
Ilyana?
Maybe we've had these discussions a lot, and so it really depends on the species you're talking about. For example, for Crocodile Hamada, there's really no big concern necessarily of moving them because populations are very large, and you're moving. Even if you move things from, say, the Gametes and the Gametes to Curacao, it's within the population. So you know, and there might still be local adaptation you're disrupting, but you know, the risk is that your translocated corals die. So, you know, it's not that it's going to have huge impacts on the population genetic structure. It might be wasted effort. You know, I guess it's perhaps the biggest risk in that case, but you know, in other species, that's a different story. Now, there would be different reasons. Like say, you know, we knew that the Puerto Rico populations would be more thermally adapted than the Curacao populations. That we could be argued contained if we move them. We do not yet have any indications this is taking place. Now, for other species, for example, the pillar coral situation is very different, where you know we're actually we just recommended trying to do ex situ crossing experiments between Florida and Curacao, because in that case we do have very strong population differentiation and very different populations. And there, you really make an argument this might actually help with this diversity in numbers, and you know there perhaps even be genetic rescue, you know, aspects to this. But we're recommending ex situ right now because, you know, if you do it land-based, there's not a whole lot of risk, I guess, but we just don't, and we wouldn't know who to ask, to be honest. You know,
that's exactly you made a perfect science answer. I was wondering to the legal and government framework how you make those decisions. And because
that's what I asked Phil in the break.
You can make all really nice arguments.
I mean, there's no issue about CITES. Yeah, if you want to export any coral material, you get a CITES, but again, that's not for reintroduction to the wild. So that's a totally different topic. And yeah, I would be really curious to. Yeah,
I have no idea either. I just know that within the Australian Ocean, there are lots of people working on the legal aspects. I don't think there's any, but that's actually, I think, a critical issue.
Absolutely.
The legal and governance aspects. If you know, so
I expect that.
Those, then
yeah, there could be something for the Nature Conservancy to look at. Yeah.
How many of the non-profit student fragmentation you know, incorporate genotyping, like within your network and also outside the operations in general improving? How often does that take place at what level, like microsatellites? Or
yeah, I'm looking at Margaret and Ilyana, but yeah, Margaret, do you want to say something?
I mean, a lot of them do. Again, there's been so much ramping up of this effort over the past 5 to 10 years. And there has been a lot, partly because of these permitting issues. Well, are we really, is it smart for us even to think about reintroducing a coral that's been in a land-based nursery or a field nursery? There has been a fair amount of thought going into how do we do this in a way that's most risk averse. We're going to move forward, but let's do it in a way that represents the least risk that we can manage. And so genotyping and tracking genotypes and using and fragmenting nurseries as many genotypes as possible has been a pretty major component of at least many of those operations. Now, sometimes there are, you know, permitting restrictions about how you can make your initial collections, and so there's limitations in different places. But it's certainly, I think, I would say, universally in the intention of all of those operations. There's probably some variation in how much investment individual operations have been able to make, but it's certainly recognized as a need and a need to incorporate as many genotypes as possible when you're doing that fragment-based propagation.
Questions?
I think there's a question online, but I can't see it on my screen.
So where's there was a laptop. Oh.
Yeah, but I'm not connected to it anymore. Check if it's. No. Okay, that's not
perfect.
Okay.
There's one about the other public aquarium.
No, where is it?
I don't have my glasses on.
Can you read it?
You don't have your glasses too. Okay. Life banks. Oh, that's something that actually came up from the public aquarium community to use public aquaria as a kind of repository to maintain live coral. an opposite to cryobanking, for example. So personally, I have a lot of doubts that this is a direction to go, because you have to imagine, I'm not sure if you have any experience with husbandry, with maintaining such systems. So at what scale, at what scale are we looking, time scale, to keep those corals alive in those aquaria? Do we look at 10 years, 50 years, 100 years? It's a huge effort. You have to, usually those aquaria that they maintain organisms from different regions, so you would have to have some security there to not get any contamination. And it's once you have a power failure, or any of your personnel does a mistake, and that happens frequently, it's all dead. It's just like that. And so there are, I think, fortunately there's no colleague here who knows better, but there are corals that have been maintained since 20 years in aquaria, but that's at a handful of institutions that have been extremely successful and have a very reliable or consistency in their team, and that's exceptional. So I don't think it's a waste of money, I guess, I'm afraid.
What about the aquarium trade? There's a lot of people in the general public who have corals in their aquarium now quite successfully. Is there any sort of analysis what that is usually maintained?
That's a good one, because what I just forgot to tell you is that what happens if you introduce, and I was working for 10 years at the Rotterdam Zoo, which has a big aquarium and a laboratory, and we were focusing on Caribbean coral. So we were like collecting every individual on Curacao, and then followed the individuals, and it was very difficult to keep track of them. And what happens, the corals that are kept in captivity for a longer time, they change their morphology. It's getting really difficult to know after a while. There's fragmentation going on eventually. And with hobbyists, what's your question?
What's the repository with the general public in their own home aquarium? There's quite a list of species that you can get.
Yeah, it's going to be a nightmare to find out what species are in private aquaria, because they look very different from what they had looked when they got into that aquarium. And so hobbyists, I think they have a very important role to be ambassadors for coral conservation. Very important to finance some of the conservation efforts through purchasing aquacultured coral, and with farms that are in places like Indonesia. But usually a hobbyist gets animals from different wholesale or different shops from different regions, so you would have a lot of contamination. Yeah.
I think we end this first session here. And
there's
okay.
Yeah, that's a great question. It happened occasionally that until recently that corals could spawn or spawned in aquaria. I have a great technology. You can simulate moonlight cycles that stimulate spawning and reproduction. That we know about that, and temperature cycles and so on. They are brooding species that have internal fertilization, and they develop larvae that are ready to settle when they are released. So that happens frequently with very opportunistic species in aquaria. And recently, a colleague called Jamie Craggs at the Horniman Museum in London really looked systematically into inducing spawning of broadcast spawning species in aquaria, and yes, he is extremely successful. He's collaborating with you at Ames. The CSIM is also, the idea is to look at inducing broadcast spawning in captivity. Yes, it's possible. And the funny thing is that Jamie, he's a curator. He manages an aquarium, the smallest aquarium I've ever seen. And he said, yeah, that's great because it gives me a lot of opportunity to do this kind of work. So he had trouble to stay awake because those corals, they spawn after sunset, they spawn in the evenings, and then with his stuff and the whole routine, it was like, it's not going to work for the things I want to do because I want to monitor those corals over months. So he just switched the light regime and they and managed to have them spawn in the afternoon after lunch break. So these studies can give really very important implications also on restoration. Yeah.